Hi students, in the last class we discussed about photosynthesis chapter, right? In that chapter we discussed about some of the basic experiments of photosynthesis process, right? So in that experiments the main theme is basic requirements for the photosynthesis process we are discussed. See, by the experimental evidences we proved carbon dioxide is required for the photosynthesis, light is required for the photosynthesis, right? We discussed one of that. There is an black paper test, black paper experiment is there, right? By the uses of the screens, and next one is the KOH experiment is there, right? Based on that, we finalized light is required for the photosynthesis process, carbon dioxide is required for the photosynthesis process, right? After that, coming to this class, we have to discuss about some of the experimental evidences, that is nothing but early experiments which are related to the photosynthesis process. Here the very first scientist name is Stephen Hales. Stephen Hales is one of the best scientists and he is also called father of plant physiology. Stephen Hales is also called father of plant physiology. He did the research on the plants and especially related to the photosynthesis process. Not only the photosynthesis, physiology means here the transportation process and the role of minerals, right? So several physiological aspects are there within the plants. Regarding to the all physiological aspects he discussed, right? So here what I am telling, okay, he given one statement regarding to the photosynthesis process. He explains about carbon dioxide and light for photosynthesis is the nourishment. See, nourishment is nothing but, see, we know, we discussed about some cells. Definitely we have to discuss about the nourishment of the cells, supplies energy. How we are supplying energy, that is nothing but the nourishment process, right? Here, the energy for the photosynthesis process. Here energy is nothing but the components are required for the photosynthesis. The components are required for the photosynthesis. So here nourishment is nothing but the total process is depends on, the total photosynthesis process depends on carbon dioxide and the light. Carbon dioxide and the light. So these are the major requirements. Major requirements. In the last class also we discussed on same points, right? And coming to the next one, Joseph Priestley, he is one of the best scientists regarding to the photosynthesis process. I thought in 10th class also we have the photosynthesis chapter. In that chapter we discussed about the Belger experiment. He did the research, okay, especially there is a duration is there 1733 to 1804. 1733 to 1804. So between that time period he did the research. Okay. And here Joseph Priestley, you are saying he is one of the best scientists. Why he is the best scientist? See, in the olden days the scientists don't have any proper equipment. In that case also they are proved by the basic experiments. So here Joseph Priestley proved that during photosynthesis process, restoration of air will be takes place. He explains the main theme of Joseph Priestley Belger experiment is restoration of air. That is nothing but purification of air done by photosynthesis process. He explained. How he explained, we are discussing now, right? See, he taken one of the Belger, or suppose he taken one of the Belger, right? See, this is one of the this is the bell jar, right? This is the bell jar. Inside the bell jar, he kept one of the burning candle, right? This is the candle. This is the burning candle, right? He kept inside the bell jar one of the burning candle, and he kept one mice, right? Suppose, okay. Yeah, this is the mice structure, right? So this is the mice. Just imagine like mice. So this is the mice. So he kept the mice inside the bell jar, and he kept the burning candle. See, after some time, the mice will be suffocated. See, lack of oxygen, lack of proper air. See, body produces the sweat. Body produces the sweat. So the mice is uncomfortable. The mice is uncomfortable inside the bell jar. So the condition is called as suffocated condition. The condition is called as suffocated condition. Initially, mice suffocated. After some time, mice will be dead. The mice will be dead. Yeah, the mice will be dead. After some time, the mice will be dead. See, what is the main motto of this experiment? See, the main motto of this experiment is burning candle. Burning candle indicates here whatever that amount of oxygen is present inside the bell jar, the oxygen is utilized by the burning candle. So mice here inside oxygen, okay, burning process, carbon dioxide is produced. Accumulation of carbon dioxide, lack of oxygen, the mice will be dead. Lack of oxygen, the mice will be dead. So that is about the first case. That is about the first case. Coming to the second case, inside the bell jar, okay, the freshly kept one of the mint plant. See, this is one of the mint plant. He kept inside. This is the mint plant. He kept inside the bell jar. He kept inside the bell jar, right? So again, as usual, there is a mice is there, right? This is the mice structure. Yeah, this is the mice structure. See, he observed so many number of hours. He observed so many number of hours, so many number of days, and so many number of weeks. But here, the mice will be survived. Here, mice survived. In the second case, mice will be survived. Why here mice will be survived in the second case? Why here mice will be die in the first case? In the first case, mice will be die. In the second case, mice will survive. Why here mice survive? Why here mice died? Why here mice died? Remember, see, once the burning candle, here whatever the candles are there, if the candles are burned, see, there is an bad air. Bad, okay, that one is indicates here bad air. Bad air indicates here lack of oxygen. Oxygen is absent. Carbon dioxide is accumulated. Carbon dioxide is accumulated. In the second case, whatever the mint plants are there, the mint plants, whatever the carbon dioxide is present, the carbon dioxide is taken and oxygen is released. Oxygen is released. So here, restoration of air. Restoration of air. In the second case, restoration of air will be takes place. That is nothing but purification of air. So carbon dioxide is taken, oxygen is released. Carbon dioxide is taken, oxygen is released. Right? That is about the second case. So based on this, we can conclude during photosynthesis process, oxygen will be released. During photosynthesis process, oxygen will be released, and carbon dioxide is taken. Carbon dioxide is taken. So this is about the main theme of bell jar experiment or Priestley experiment. All of you remember, by the Priestley experiment, we can conclude during photosynthesis process, restoration of air will be takes place. That is nothing but purification of air will be takes place. right? Some other experiments also there, we'll discuss now. Here, coming to the next scientist is the engine house. See, we are very familiar with this experiment. His time period is 1730 to 1799, right? So he did the research on hydrilla plant. We know about one of the glass beaker he taken, right? This is one of the glass beaker, and there is in water, right? Inside the glass beaker, pour the water. After that pouring process, right? See, he collected some of the hydrilla plants. He collected some of the hydrilla plants. So suppose these are the hydrilla plants, right? So these hydrilla plants especially kept this is the glass funnel, right? The hydrilla plants kept inside the glass funnel. The hydrilla plants kept inside the glass funnel. After that, upper side of the funnel, he kept one of the test tube. Exactly not like this. This is one of the hydrilla experiment. I thought most of the students are familiar regarding to this experiment. He collected some of the hydrilla plants. Okay, the plants kept it inside the glass funnel, right? Whatever that opening part is there, narrow opening part of the glass funnel, okay, is covered with the one of the test tube, right? So total this equipment, see, this is the total equipment, and pour the water inside the beaker, right? So the total equipment is covered by the water. So again, here two conditions are there. See, one is absence of light and the other one is presence of light. See, if the total equipment is exposed to the sunlight, that is the one case. Other case is absence of light. So first one is here, absence of light. In this case, no air bubble formation. See, whatever that hydrilla plants are there, the plants are not producing any air bubbles. Sir, why here air bubbles are produced? We know during photosynthesis process, the byproduct is the oxygen. Once the total setup is exposed to the light, all this is the glass material. Easily light passes and reaches the hydrilla plant, and there is a chance for the photosynthesis process. Actually, these are the hydrophytes. Hydrilla plant, that is the hydrophytes. See, that's the reason why there is no need to supply the carbon dioxide. Even these plants have the capacity to absorb the carbon dioxide from the water. Because these plants are hydrophytes which are grown under the water, right? So here, see, if the total equipment, if we kept presence of sunlight, or if we supply the sunlight, right, in that case, see, whatever that the green parts of the hydrilla plants are there, the green parts produces air bubbles. See, the production of air bubbles is nothing but releases the oxygen. So here, this is the plant. So two conditions are there. See, one is that absence of light, there is no air bubbles formation. Other one is the presence of light. Okay, definitely the air bubbles formation takes place because during photosynthesis process, oxygen is produced. So this is one of the evidence. So how can we conclude the produced gas is the oxygen? Simple. See, if we kept any fire at the opening of the tissue, okay, that is light and high, okay, that one is brightly lightened. That fire material brightly lightened because oxygen is supporting for the burning or firing. Oxygen is supporting for the firing. So that's the reason why. See, if the carbon dioxide is there, the carbon dioxide is not supporting for the firing. The carbon dioxide is not supporting for the firing. So based on the property of the gas, we can conclude it during photosynthesis process oxygen is liberated. During photosynthesis process oxygen is liberated. So that is about that hydrilla experiment done by the engine house. And other one is that Engelmann, T.W. Engelmann, he is also one of the best scientists, and he is a first scientist. Okay, photosynthesis occurs in specific light. See what I am telling here. Whatever that photosynthesis process is there, the photosynthesis process. Okay, at particular light regions, maximum rate of photosynthesis will be occurred. Maximum rate of photosynthesis will be occurred means we know about the sunlight. If we subjecting the light is subjected for the subjected to the spectrum. See, the light is divided. The light is divided into that vector. So in all the regions, okay, the photosynthesis rate is not equal. The photosynthesis rate is not equal. So at different regions, the rate of photosynthesis is different. The rate of photosynthesis is different. So he first did that the experiment in certain regions, maximum rate of photosynthesis is there. Okay, he identifies. So here, Engelmann, he used the spectrum to split the light. After splitting process, the light illuminated on green algae and cladophora. He used that green algae, okay, cladophora, right? So after that illumination process, the total setup is mixed in a suspension, especially that suspension. Remember, okay, having the aerobic bacteria. So what is the use of aerobic bacteria? Aerobic bacteria provides the carbon dioxide. Aerobic bacteria provides the carbon dioxide. Simple spectrum, splitting process, illumination on green algae. Here other one is the cladophora, right? See the total setup kept in the suspension. The suspension contains the aerobic bacteria, because aerobic bacteria provides the oxygen for photosynthesis process also. Aerobic bacteria provides the carbon dioxide for photosynthesis process also. Carbon dioxide is required for photosynthesis process. Carbon dioxide is required. So that's the reason why that aerobic bacteria provides the carbon dioxide, right? See, after the total setup, after some time, whatever that green algae and cladophora, right, whatever that aerobic bacteria is there, all these are majorly accumulated at the red light and the blue light. Within the spectra, different lights are there, vector is there. Within all these regions, mainly these organisms are accumulated within the red and blue lights. So here what indicates that accumulation process within that region, maximum rate of photosynthesis will be occurred. Within that red region, within the blue light region, maximum rate of photosynthesis will be occurred. So that is about that Engelmann experiment. So here the Engelmann used the spectrum for the splitting process. Here the green algae and the cladophora. After that, light illumination process kept in a suspension. The suspension contains the aerobic bacteria. So majority of these are accumulated within the red light and the blue light region. So that one indicates majorly, okay, photosynthesis occurs within the red light and blue light regions. So that is about the Engelmann experiments. Next one we will discuss about the next two scientists. Coming to the next scientist, next two scientists here. One of the Robert Hill, okay, he is one of the best scientists. He explains regarding to the photolysis of water. So presence of light energy, there is an lysis of water molecules that takes place. That lysis process is supporting for the evolution of evolve the oxygen. Whatever that water molecules are there, the water molecules are split into H plus ions, and the oxygen evolving occurs. So here there is in chloroplast. See, within this experiment, the chloroplasts are completely separated from the cells. So isolation of chloroplasts, which are kept inside the tissue, right? See, the chloroplasts are isolated from the Stellaria. The plant name is the Stellaria, right? That is the plant. From the plants, the chloroplast isolated, okay, which are kept inside the tissue. Here, potassium ferrocyanide and potassium ferroxylate. Potassium ferrocyanide and potassium ferroxylate. Both the reagents are added, right? These reagents are called as Hill reagents. These reagents are called as Hill reagents. This reaction is called as Hill's reaction. This reaction is called as Hill's reaction, right? So this is one of the Hill's reaction explains regarding to photolysis of water, and the main concept of photolysis of water, evolve the oxygen, right? So here oxygen releasing is occurred by the presence of chemical substances and the chloroplast, right? So that is about that main concept of the Hill's reaction. This reaction done by the Robert Hill. So on his name, okay, the reaction is given as, okay, Hill. So Hill is his name. The reaction is also called Hill's reaction, right? Coming to the J.V. Sachs, he is also one of the best plant physiologists. He did the research on the photosynthesis process, and he explains that how the glucose molecules are synthesized, right? The basic explanation is given, not complete and not the structural explanation, but the basic explanation is given by the J.V. Sachs. The glucose production, right? Here there is a light requirement is there, carbon dioxide requirement is there, light requirement is there, carbon dioxide requirement is there. Both these are very important, right? And glucose molecule synthesis will be takes place, right? Here the glucose is converted into the starch. The glucose is converted into the starch, and he explains for the synthesis of glucose, and he explains for the starch formation, and he also concluded starch is the storage form of carbohydrates. Starch is the storage form of carbohydrates within the plant body. So J.V. Sachs explains about that the the glucose synthesis. For that glucose synthesis, carbon dioxide is required, light is required. He explains starch formation takes place. The starch formation, okay. The storage form of carbohydrates within the plant body is the starch. That is about that some of the early experiments, and one more experiment is also there. We'll discuss in the next class.